So I talked a lot about RNA sequencing, proteomics, and all of that. What does this really mean? And why do we invest so much of our funding into these high throughput techniques? So I'll talk about histology, what that tells us, immunohistochemistry. So immunohistochemistry is a very basic thing that is done as a part of clinical research and also at preclinical research. What histology or immunohistochemistry tells us, and what this told us from this study, is the morphology of the cells. How does the cancer look under the microscope? What are the markers or the proteins that this cancer expresses, which can be used for diagnosis? And what are the specific markers that, in our case, small cell lung cancer expresses that can tell us a lot about the disease when we try to identify drugs? And it also tells us that this cancer is actually very heterogeneous. When we talk about NGS or whole exome or whole genome sequencing, what that tells us is about the high mutation burden, what are the genetic alterations that can be targeted, what are the genetic alterations that cannot be targeted. And this told us that small cell lung cancer lacks a lot of the recurrent genomic alterations that can be targeted. But for non-small cell lung cancer, these studies told us that there is a KRAS mutation and an EGFR mutation that could be targeted. Proteomics, it again tells you a lot about the proteins, and it tells us about the drugs that can be targeted. And this is a lot of the study that we did, is we found through proteomic analysis that some of the drugs targeting DNA damage response pathway could be effective targets. Finally, RNA sequencing tells us the subgroup of these tumors and also helps us understand some of the targets that are currently in the clinic. You may identify DLL3, you may identify PARP and other targets that are currently into the clinic. Finally, single cell RNA sequencing. Why do we need that? Because we need to understand rare population of cells. And if you consider bulk RNA sequencing like a smoothie, we really want to understand the fruits that went into that smoothie. And this told us a lot about small cell, about the heterogeneity, about the immune microenvironment, and about rare populations. So with that, I'd like to, sort of bring all this together, because I know I took you through like a lot today, but I would really want to break it down, because basic science, we invest years and years of our life into understanding the biology of the disease so that we can take targets into the clinic. And it really transforms cancer care. Breakthroughs happen only when waiters and you, patients and caregivers, we join forces and decode the mysteries of cancer. There's a complexity to the drug development process. It cannot be hurried, because we have to identify new targets, we have to take new molecules, and the deeper understanding of cancer biology will lead to better treatments. Life scientists, we are the partners in innovation, so talk to us. From bold ideas to real-world impact, that's what we invest our time in. Researchers drive the future of cancer care, because with every discovery, we hold the potential to change lives, which becomes a therapy for tomorrow. What do we need? We need cutting-edge infrastructure, because it is a foundation of understanding cancer. We need resources, we need funding, we need partnerships, we need perseverance, most of all. Years can go into understanding that. State-of-the-art technology, I spoke about some of them, I'm happy to answer more questions, but it accelerates discovery in an unprecedented space. And then you need brilliant minds to come together. We need to have open conversations like this, where we all come together into finding new therapies. I want to thank everyone, all my team members, but innovation and research saves lives, and the future of cancer treatment is being built today. And with your help, the future looks really promising. With that, I'd like to thank you all, and I'm happy to answer questions in the Q&A session. Thank you.